Chapter 21's topic is the study of the immune system, which, as we know, provides us with resistance to all the different diseases that our body comes in contact with. It's made up of two intertwined systems. These two systems are detailed in chart 21.1 in your text. You see here innate and adaptive immune system. So the innate immune system, remember, is non-specific, meaning it doesn't care what the foreign antigen might be. It's going to react the same way, whereas the adaptive immune system is specific, meaning we have to have exposure to a certain type or a certain antigen for us to produce immunity for that specific antigen. And only when we are exposed to that specific antigen can we create immunity towards that. One of the downfalls of the adaptive immune system is that it is time sensitive, so it takes longer, whereas the innate immune system does not really take time. So you see here some of the things, some of the barriers and defenses that we have in the body. Surface barriers being the first, what's called the first line of defense. So these are going to be our skin and our mucous membranes. The second line of defense are all of these listed structures here. So we have phagocytes, natural killer cells, inflammation, antimicrobial proteins. We're going to focus specifically on a structure, a protein structure called the interferon, and then fever. For the adaptive immune system, you might have heard of these types of cells. We have T cells and B cells. If we recall from our study of the white blood cells, these two cells are lymphocytes. So in the secondary line of defense, we have phagocytes. Phagocytes operate via phagocytosis, which is the process of consuming something, whether it be, as it says here, a pathogen or cellular debris. It could be just dead tissue. So in step number 1, you see adhering to whatever this pathogen is. Typically when you see a green rod-like structure like this, it's going to depict bacteria. In step number 2, you see that the engulfing is forming what's called a phagosome, which is a membrane-bound vesicle. And then you see the lysosome. If you recall from our study of the cell, the lysosome will then fuse with the phagosome and release its enzymes, which you see all spotted throughout here. And these chemical enzymes will then dissolve, so you see toxic compounds and lysosomal enzymes destroy/dissolve the pathogen, and then exocytosis may take place to remove the indigestible or residual material. Sometimes we can use some of this material, but what we don't use, the residual, is typically discarded. Number 2 type of second line of defense cell is called a natural killer cell. Natural killer cells are different from phagocytosis. They're non-phagocytic, therefore they do not destroy cells by engulfing them. What they do is they attach themselves to a cell that might be infected with a virus or a cancer. Remember, both of these types of cells have to feed on something. They have to have a host. Therefore, a cell that is infected with these two types of things needs to be killed off before the virus or the cancer can spread through its host. So the process is called apoptosis, and those of you that have taken bio, or you might remember from the study of the cell, apoptosis is an induction where, I should say, the natural killer cell induces the infected cell via apoptosis to kill itself. So basically it will secrete chemicals that enhance inflammatory response and cause this cell to undergo a process where it commits literally cellular suicide. And that's good, that's ideal, because then it kills whatever virus or cancer cells might be inside of it. Inflammation is a very important process that we have in our body that's triggered when our tissues have some sort of injury. this injury could be like an accidental, as you see in this picture, a nail through the skin, so trauma. It could be burning, it could be chemical irritation, or it could be infection. So in this case, we have the opening of the skin with the nail, and now we have some sort of microorganism that's come into the body that we now have to take care of. So there are many benefits, as listed in your text, of inflammation. You can go through those benefits. I want to make sure I point out the cardinal signs of inflammation. So there's really four signs or symptoms of inflammation. So redness is number one, heat is number two, swelling or edema is number three, and pain is number four. And sometimes when this is so bad, you can have impairment, which is sometimes considered number five. Impairment. We've all had probably pain to the point where we had an impairment, even if it means we can't, turn our head when we're driving, or we've hurt our arm, we can't write properly, we tweaked our ankle, we can't do things. So the importance of these things is all due really to blood flow. Redness is due to increased blood flow to an inflamed area, as is heat. So when there's more blood, it's going to turn red, it's going to feel warm, there's going to be more fluid, which means that it will be swollen, and there might be pain of the injured tissue itself, but also if there's swelling, there could be a nerve that's being compressed, and that could cause extra pain in addition to the injury of the tissue itself. So what we're looking at in this picture are the four steps of inflammation. So for the phagocytes to be mobilized, there has to be a series of events that takes place. First thing that happens is that leukocytosis occurs, which means, if you break it down literally, the movement of white blood cells, so white cell process. Bone marrow releases the lymphocytes or leukocytes that will respond to injured cells releasing chemicals. So it says up here, inflammatory chemicals diffusing from the inflamed sites act as chemotactic agents, which means that the chemicals will attract the white blood cells to the inflamed area. Remember that these things don't have noses, they don't have cell phones, they can't call each other, they have to act via chemicals. The second thing that happens is once the white blood cell down here gets close enough to the area, it clings to the wall, so margination. Remember that this should not normally happen. We normally don't want things clinging to walls in blood vessels, especially in capillaries, because it can block them. This is, however, the ideal case, because once margination takes place, step number two, they squeeze through the pores of the capillaries. Remember that capillaries are normally very porous, meaning they have holes. So the white blood cells are able to squeeze through here. Maybe this is a neutrophil. Squeezes through the wall via what's called diapedesis, which means literally through foot process. So it's the process of gaining feet. They can walk out and into the tissue space. Number four, chemotaxis, is following this chemical trail. So they literally sniff out the chemicals, following the trail to the area of injury. Number four on this list of secondary or second line of defense is what's called an interferon. An interferon is a protein. What's going on in this picture is that a virus has entered, step number one, a virus has entered a cell. When the virus enters the cell, ideally what happens is, in step number two, an interferon gene is switched on in the DNA, meaning the DNA is going to light up and produce a messenger RNA. Remember the whole process here of DNA to RNA. Messenger RNA is to produce a protein. So it's going to turn on a switch where a messenger RNA that codes for a interferon molecule is produced, and then this messenger RNA will go out into the cytoplasm where it will meet up with the ribosomes to form a very important protein called an interferon, which we see here as step number three, producing the interferon. So that's all fine and dandy, but what has to happen is through exocytosis, the interferon will then travel to the neighboring cells, the healthy cells, receiving that interferon will then turn on its DNA to produce an antiviral messenger RNA, which will then go out into the cytoplasm to produce a mechanism to block this virus from being able to enter the nucleus of the cell. Therefore, the virus cannot inject its genetic material into the DNA, and it won't be able to replicate. So basically, the gist of this concept is that an infected cell, okay, will warn healthy cells that are neighboring it about the impeding danger. So in this case, a virus. So it's kind of like your neighborhood watch. You've got one person who calls everyone and says, hey, look out, there's something shady going on. The last piece of this second line of defense is a fever. So abnormally high body temperature that is systematic response to invading microorganisms. In this case, we have the leukocytes and the macrophages that are in the exposed area secreting a substance called pyrogen. So pyrogen is a hormone that's going to travel around the bloodstream, but the only place it's going to act is on the hypothalamus. If you recall from Anatomy 1, the hypothalamus is the body's thermostat. So when the pyrogen hits the hypothalamus, the body temperature, okay, will rise. And there's benefits to a fever. We want to have a moderate fever in certain cases because it speeds up our metabolic rate, increasing our tissues repair, but it also sequesters, which means it holds back certain minerals that microorganisms need to survive, such as iron and zinc. So the liver and the spleen are smart. They hold back these things to try to starve out the microorganisms, and then obviously they will die and can't replicate. These mechanisms are all detailed, by the way, in a chart 21.3 in your text if you want to take a look at it. The next part of this presentation details the adaptive immune systems. The adaptive immune system is specific, and it can eliminate many different things in the body. It works in conjunction with the inflammatory response and activates the complementary system, such as the proteins that we just mentioned. However, it does take time, so it is time sensitive, like we said. So it's very specific, and it also has what's called memory. So if we think about, I'm going to write it over here. Immunity has one of the basis concepts of our immune system is that we do have immunological memory, meaning we can mount stronger attacks on second and third and fourth and every subsequent exposure to certain substances. Now, there are two branches of our adaptive immune system. You'll see them listed up here. We have humoral immunity and we have cellular or cell-mediated immunity. These two systems are very important. The humoral system is based on antibodies, whereas the cellular system is lymphocytes themselves. So when we talk about humoral antibodies, humoral, as you recall, means liquid, such as the aqueous or the vitreous humor that we found in the eyeball. So we're talking about watery substances of the body, whereas cellular immunity obviously refers to the cells themselves, such as cellular targets, which are targeted by the specific lymphocytes. So we know that we have to be exposed to a certain antigen. Remember, an antigen is something that provokes the immune system. Something that's either foreign, or if we have autoimmune, that can be a problem, or cancerous cells. So these antigens will provoke and promote a response by the immune system. With the antigens, they have certain chemical shapes. They're very complex chemicals. Your book chooses to detail on in figure 21.7 an antigen with three different antibodies, antibody A, B, and C. One's shaped like a triangle, one's shaped like a circle, and one's shaped like a square. These are very elementary. It's a very elementary picture. The actual antigenic determinants in your body are very complex. You'll see this further down in the chapter. They're very complex shapes. They're not just like kindergarten square, circle, triangle. The point of this, however, is to note that each antibody has a specific binding shape that's complementary to the antigen, so it's all based on the chemical shape. We are exposed to millions of different things almost every day, foreign to our bodies, and these things, if you imagine, all have different shapes, meaning there's all different types and different classes of antibodies that will complement these foreign antigens. If you take a look at section 21.4 in your text, it's going to start to detail the B and the T cells. B cells are used for humoral immunity. T cells are used for cellular immunity. Then we have a third type of cell called an APC, or antigen-presenting cell, which are not responding to specific antigens, but they're playing auxiliary roles or helper roles in immunity. So in this chart that you see in front of you here, we see the process involved in lymphocyte development. It's going to be an important process to know. You see here primary lymphoid organs versus secondary. Primary organs are the red bone marrow and the thymus. So those are the ones that are listed in this particular step right here. So step number 1 is the origination of B and T cells. Lymphocytes are all formed in red bone marrow. One of them will, however, migrate to the thymus. This is the T cell. After formation, it will go to the thymus gland to mature. The B cells, however, will remain in the bone marrow for maturing. When we say maturing, they are developing, as you see this word here, immunocompetence, meaning they are becoming immune system smart, literally. They are learning what their job is, and they're learning what it is they need to be doing in the immune system. And they will also undergo what's called self-tolerance. If they don't pass their tests, they undergo apoptosis and they die, which is very dramatic, considering if you guys didn't pass your test, hopefully you won't die. So thank God we don't undergo self-tolerance. Step number 3, once they gain their immunocompetence, they're still naive. So in this case, this means they have not been exposed to the actual antigen yet. They've just learned about it. It's kind of like when we learn about school, we learn how to become a nurse, we learn the book stuff, but then we have to go practice in clinics. We have to go be exposed. So the same thing happens here. They have to be exposed, and they do so by going to a secondary seeding site, which is typically the lymph node or a spleen maybe. And in this, they will hopefully encounter the antigen. When they encounter the antigen, step number 4, they will receive the antigen in their binding site, and they will become activated. Once they become activated, step number 5, they are able to proliferate. Remember, the word proliferate means to multiply rapidly, and differentiate, meaning they can either turn into an effector cell, which we'll talk about what the options are there in a minute, or they can remain and become memory cells. And remember that the memory cells are the basis of our immunity. So that's going to be a very important concept. Once they have their specific function, effector or memory, they can then circulate through the system and throughout the secondary lymphoid organs. If you want to take a look at an overview, table 21.4 in your text gives you a pretty good overview of what the differences are between the B and the T cells. The third type of cell is an antigen-presenting cell. Now I wrote on here figure 21.10 in your book shows you what a dendritic cell looks like, and I wrote scary just because it looks kind of like a gnarly cell. It has all these dendritic processes. It kind of reminds you, if you think back to the dendritic tree of the neuron, the input portion of a neuron. This is how it gets its name, dendritic cells. It's commonly found in connective tissue in the epidermis. These are all the boundary tissues, meaning they're going to be exposed. These places will be exposed, have a higher percentage of exposure to foreign microorganisms. It can phagocytize the pathogens, and then present the pieces. So what it does, since you see antigen presenting cells, it presents the fragments to the T cells. Basically, it gives it to them and says, hey, here, learn this. This is a presenting, I need you to take this piece and I need you to learn it so that in the future, if we are exposed to it, you can produce immunity to it. So it's a very key link between the innate and the adaptive immune system. Now, B cells, okay, so B cells, when they are exposed to a target antigen, it provokes, like we said, a humoral response. Antibodies that will be specific for that particular antigen are then produced. So B cells are activated when antigens bind to their receptors on their surface. When this happens, it can lead to proliferation and differentiation into the effector cells. So the effector cell for a B cell is called a plasma cell. You need to know this. Okay, a plasma cell. And plasma cells are the ones that will secrete and produce and secrete antibodies. As detailed in your text, specific antibodies can be created at a rate of 2,000 molecules per second for 4 to 5 days, their lifetime, and then they will die. These antibodies can circulate then in the blood or the lymph, which will bind to free antigens, so bind to the foreign substances, either marking them for destruction or by other mechanisms, which we'll look at the different mechanisms that happen when an antibody forms with an antigen. Okay, so if you look at figure 21.11, and that's, I think, a part 1 and a part 2 for it, I want you to go ahead and pause this right now and look at this. Find this in your text, please. Okay, now that you're there, please take a look at this primary response image, and we can see here an initial counter. So the initial encounter with the antigen is this little red piece, looks almost like a little flower, and it's reacting with that middle B lymphocyte, so B cell. It's not reacting with the other ones because they are not immunocompetent for that specific antigen. It's only going to react, remember, it has high specificity. Once that binding takes place, it can then proliferate to form a clone, a copy. These clones can then become either a plasma cell, which you see on the bottom left. Those are going to be your effector cells, meaning they were going to have some kind of effect or response, which is secreting antibodies. Or, I'm going to write on the screen here, because our choices are effector or number 2, memory. So some of these cells will remain and remain behind and become what are called memory cells, meaning that if that antigen encounter happens again, so if we're re-exposed to the same thing again, that memory cell remained and it can provide a immune response. So if you now look over at the second part of this image, that one little memory B cell that remained can now, and as it says in the secondary response, could be years later, creates clones, so clone of cells identical to ancestral cells back in the first step, and then the subsequent challenge by the same antigen results in a more rapid response. So the same antigen now is encountered again, and now look, we've actually produced more plasma cells the second time around with more antibodies and more memory cells, meaning that a secondary response versus a primary response, a secondary re-exposure to the same antigen provides a faster, prolonged, and more effective response because of this memory that we had. So as you see in your text, it can be a response within hours, not days. The antibodies will peak in 2 to 3 days, not 7 to 10 days like the first response. And it spares us, hopefully, the symptoms of the infection, which is the main, so that's like the main goal of it, is to not have that immune response where we feel ill, our immune system is not overtaken by the antigen primarily. Okay, so this brings us to active and passive humoral immunity. if you're still in your book, you can turn, and it's figure 21.13 is what we're looking at here. Humoral immunity, active and passive. So with these two branches of immunity, we have two types of active humoral immunity. Active means that we will produce actively, so actively produce the antibodies. Whereas passive means that we are given the antibody. It is passed to us. So the specific ways: natural, acquired, active immunity means that we had direct exposure to a live pathogen, and then we produced subsequently our antibodies. An artificial acquiring means that we are given a vaccine, which is either a dead or fragmented pathogen, and then we will actively produce antibodies. A naturally acquired passive type of immunity means that we are given antibodies from our mother via the placenta, so that's obviously before birth, or then after birth via breast milk. So it's one of the advantages, one of the pluses of breastfeeding is passing natural acquired immunity in the form of antibodies through the breast milk. Artificially acquired is an injection of what's called exogenous antibodies. These typically aren't options for everyone. Typically these are done in cases where people have immune deficiencies or severe infections such as hepatitis, and it's even done, as I recall, for kidney donors to make sure that there is compatibility regardless of the type of blood that the donor and the recipient have. Typically the blood is matched. Now, these passive immunity types, remember, since they're passive and you're not actively creating them, once those antibodies are used up, they are gone. You're not producing any more. So your protection ends when the antibody is either used up or when it degrades due to old age. So these antibodies are obviously an important aspect of our immunity. The last part of this presentation details T cells. T cells are more complex in their classification and their function. We see that the T cells are broken down into their differentiated types by a glycoprotein on their surface, so the surface of the plasma membrane. And so these are called CD4 and CD8, which you see in the chart here, CD4 and CD8. These two precursor cells will then eventually become either a T cell, helper T cell, or a T regulatory cell, or a cytotoxic T cell, which I've listed over on the right. So these specific types of cells obviously then have different functions. As given the name helper, the CD4 cells become helper T cells, which will activate other B cells, other T cells, macrophages, and they also, these CD4 cells might become a regulatory T cell, which is a immune response moderator, or they could remain as memory cells, whereas the CD8 cells become the cytotoxic T cells that are the more destructive types of cells. So the naive cells are called CD4 and CD8. Please make sure you remember that, especially this one. We're talking about a debilitating disease called HIV, which you guys have probably all heard of. That is a CD4 disease. So just like we saw over in the B cell chart, there has to be immunocompetence and there has to be activation. You can see in this picture the APCs are presenting, there's class 1 and class 2, they're presenting the antigen to the CD4, the naive CD4 or CD8 cell to become activated. And once they become activated, there are some that remain as memory cells, and then we have down here the effector cells. Cytokines are a chemical class that play an important role in this process because they are chemical messengers of the immune system. They will be responsible for the development, differentiation, and how we respond. You might have heard of interleukins, and interferons also play a role in this system. So table 21.7, you can see all the different types of cytokines which are important for immune response. T helper cells are going to be super important because they are adaptive immune response role activators. So they activate both the humoral and the cellular arms of the immune system. Without T helper cells, there is no immune response. So if you don't have T helper cells, you can't mount attacks against foreign antigens. So when they activate B cells, obviously then that stimulates them to produce, to divide, proliferate, and begin antibody formation. They also help to activate CD8 cells. They stimulate the CD8 cells to become cytotoxic T cells, which is important. Not only that, but the T helper cells also go back and help to amplify the innate immune system. So they can activate macrophages, they can activate the neutrophils to become phagocytic, they can mobilize lymphocytes. It's very important for T helper cells to be able to activate and mount immune responses. So the cytotoxic T cell directly attacks and kills other cells. They can do so, the cells that they target, before we get there, virus-infected cells or bacterial or parasitic cells, intracellular or extra, also cancer cells and foreign cells. And how they do so is they release chemicals called perforins or granzymes. Perforin is going to create a hole, literally, in a cell. So if you're looking at figure 21.19, I'm looking at A here. This is an important picture. It shows you how the T cell literally latches onto its target mechanism, and it will create these pores called a perforin, and then it will release these granules that have enzymes that come inside and stimulate apoptosis of that target cell. And if you look furthermore at figure 21.19b, this shows you an actual micrograph of the cytotoxic T cell latched onto a cancer cell. It's a pretty cool picture. So then we have our regulatory cells. Regulatory T cells are important because they try to regulate our systems that doesn't get out of control. So it's very important in preventing autoimmune reactions, suppressing self-reactive lymphocytes, and so we're learning how to use them and learning how to induce tolerance to a transplantation issue. So they're very important, and they're often very studied in this day and age. Moving on to organ transplants and rejection. Before we get into that, there's four really types of tissue transplants that we undergo. We undergo autografts, which is, that's basically self. So taking skin from one place and putting it somewhere else, or taking an artery or a vein for bypass surgery, or even bone grafting. So that's self. So it's obviously no risk of rejection there because it's your own tissue. Now, isograft, only a very small percentage of the world's population will be eligible for an isograft, and that's because you have to have an identical twin. So the success rate in isografts is pretty high because you share over 99% DNA, so identical twins. But me, for instance, I don't have an identical twin, so I'm not even eligible for this one, so I can cross that one off my list. Now, a xenograft is going to be an animal part that is put into a human. So we can see this used in the cardiovascular system with valves. We also see this in the skin. And then the last one, which is the most common type for humans, is an allograft, and that is the same species. So for us, obviously, human to human. So we see kidney, we see different organs, we see heart, lungs, all kinds of transplants take place. And this is called an allograft, transplant from the same species. So before this can all take place, histologists have to compare tissues. They have to match blood types, match blood antigens, match what are called major histocompatibility complex antigens. So once it's determined to be a match, then typically the surgery is completed. Afterwards, however, you will be most likely given immunosuppressants in order to prevent your immune system from attacking your new organ or your new tissue. This is obviously dangerous and has side effects, which are other types of infections. Since you have effectively suppressed the immune system, you might be more susceptible to other diseases. Highlighting, lastly, some of the diseases that can affect the immune system. First is called SCID. It stands for severe combined immunodeficiency. And in this case, it's a genetic defect where your B and your T cells are affected. They are defective, I should say, and they are so defective that you are susceptible to most every bacteria/virus out there. So this can be a fatal disease for people. It's treated with bone marrow transplants and with other types of medications. You guys might have seen this depicted on like TV shows. They put people in bubbles, so you might have seen SCID before. Now, Hodgkin's is a type of lymphoma. There's Hodgkin's lymphoma and there's non-Hodgkin's lymphoma. It's basically the presence of a certain type of cell called a Reed-Sternberg cell for Hodgkin's lymphoma. Acquired immunodeficiency. This is literally a cancer of B cells. So we see this in the lymph nodes typically, and it leads to immunodeficiency. Now, HIV and AIDS is a big one. HIV, as we know, is transmitted through bodily fluids. So you've got your blood transfusions, you've got sexual intercourse, sometimes even oral sex if there are open sores and there's mixing of fluids. Mother to fetus, blood-contaminated needles. So you guys go back to the 5th grade and learn how this particular thing can happen. Just kidding, by the way. Don't go back to the 5th grade. HIV is dangerous because it affects your CD4 count. So remember, think back to what CD4 was. CD4s were your naive T helper cells, and we stated that if you do not have proper T helper cells, you cannot mount an immune response. So CD4 receptor cells are affected by this disease. Initial symptoms of HIV contraction, typically flu-like symptoms. However, if you Google HIV symptoms, you'll see that many patients report no symptoms, which is scary because that means they might not properly seek testing and treatment. If they don't know that they have the disease, they could furthermore spread the disease. That's obviously very dangerous. HIV suppresses the immune system initially. Then you go through a period, typically 10 to 25 years, of asymptomatic period where you won't feel many symptoms of the disease. However, it is lying dormant and it begins to multiply during these years of dormancy. Now, the difference between the 10 and the 25 years is typically medications. So if you're taking your antiviral medications, you extend this period of latency twofold. So, I mean, if you tell someone 10 years versus 25, obviously they would want the 25. Now, once the HIV progresses, then it becomes what's called AIDS. So one of the common misnomers I hear about people is that they say, don't, catch AIDS. You don't catch AIDS. You are infected with HIV, and then the HIV leads to AIDS after 10 to 25 years. So AIDS is obviously then a lot worse. It's when the immune system is completely crippled. It's when your CD4 counts are really, really low. So we have very low CD4 counts. And so There's severe weight loss, there's night sweat, there's lymph node swelling, there's a specific type of pneumonia, there's a growth on the skin, a cancer of the skin called Kaposi's sarcoma. Typically, these patients who have AIDS are going to die of a simple infection. So the AIDS doesn't actually kill you, it just cripples your immune system to the point where you can't fight off simple diseases. And the last one here, autoimmune, is obviously autoimmune means self. It's a self problem. So the immune system cannot recognize which cells in the body are self and which ones are foreign. So it starts to attack healthy tissue. There are some examples in your text. You might have heard of some of these: rheumatoid arthritis, myasthenia gravis, multiple sclerosis, Graves' disease, SLE. So there's a whole class of autoimmune disorder, and we're not going to go into the specifics of them, but you can look through and learn about how they're treated and whatnot as well. So this concludes our talk on Chapter 21, the immune system. I will be posting more as the semester goes along.